Okay, hello, my name is Christine. First of all, I would like to thank the organizers for letting me present my work today. I'm a PhD student at the Novo Nordisk Life in Vivo Pharmacology Center, and for my case it's a collaboration between Department of Immunopharmacology at Novo Nordisk and Department of Veterinary Disease Biology at the University of Copenhagen. The title of my presentation is Piroxicam-accelerated colitis in the interleukin-10 knockout mouse, a new model of human inflammatory bowel disease. And at the Department of Immunopharmacology at Novo Nordisk, we work with in vivo models of chronic autoimmune diseases in humans. And these studies are for proof of concept, mode of action, etc. The hypothesis of this project is that the Piroxicam-accelerated colitis interleukin-10 knockout model could function as a tool in the preclinical research and development of new therapeutics against inflammatory bowel disease, known as IBD. In order to study this hypothesis and assess the translational value of the model, I needed to do characterization of its clinical features, the underlying pathogenic mechanism, as well as study the model's ability to respond to existing therapies against IBD. IBD is a chronic relapsing inflammatory disorder in the gastrointestinal tract of human. The disease is divided into two distinct disorders. The first is Crohn's disease, in which the lesion can affect the entire gastrointestinal tract. This disease is characterized as being a transmural focal inflammation, and we have development of granulomas, strictures, and fistulas. There is no cure for Crohn's disease. On the other hand, we have ulcerative colitis, in which the lesions are restricted to the colon and the rectum. In this case, we have superficial, the inflammation is superficial and continuous, and colectomy can be curative for some patients with ulcerative colitis. For both of these disorders, there is a huge unmet need for new effective therapeutics. The etiology of IBD is still unknown, but it is proposed that the development of IBD requires the presence of three factors, that being a dysregulated immune response, a defective mucosa barrier, and bacteria in the gut lumen. So, the model that I work with is based on the knocking out of the interleukin-10 gene. Interleukin-10 is an important immunoregulatory cytokine which has a broad anti-inflammatory property. In 1993, Kuhn showed that interleukin-10 knockout mice develops a chronic enterocolitis. However, in the stable at Novo Nordisk, the incidence of spontaneous colitis in these interleukin-10 knockout mice, based on a black 6 background, is very low. And that, of course, limits its use in medical research. Luckily, the disease can be accelerated by administration of Piroxicam. Piroxicam is an NSAID which inhibits the production of prostaglandins in the gut. Since prostaglandins is very important for the growth and maintaining of the gut epithelium, treatment with Piroxicam causes a decreasement in the mucosa integrity. The combination of the lack of IL-10 and the treatment with Piroxicam causes an immediate severe development of colitis in these mice. Here you see the protocol of the model. I give Piroxicam to interleukin-10 knockout mice from day 0 until day 9 of the experiments. Then I terminate the treatment and follow the mice for 2 more weeks. And then I look at the clinical features of the mice and compare it to what we see in humans. Human IBD patient has weight loss and they have diarrhea. Here you see the manifestation of the model. We have present weight change and feces softness. And you can see that the PAC IL-10 knockout mice develops weight loss and diarrhea compared to interleukin-10 knockout controls and wild-type controls. The disease peaks at day 9 of the experiment. At that day I terminate Piroxicam treatment, and you can see that the disease ameliorates a bit afterwards. Then human patients also have raised CIP, which is an acute phase protein. In mice we have another acute phase protein called haptoglobin, and you can see that the model has significant increasement of haptogen level, in particularly at day 7 during Piroxicam administration in the acute phase of the disease. Then the level of haptogen declines a little bit at day 21. Then I wanted to look directly at the colonic mucosa, and I did that by endoscopic analysis and histology. I hope it's okay, it's quite clear here that here you have the wild type mice, and here you have a Piroxicam-accelerated IL-10 knockout mouse. And I hope it's clear that the model develops a thickened, non-transparent mucosa which has a granulomatous appearance, and we have a disappearance of the vascular in the mucosa, vascular pattern in the mucosa, and you also have some ulcers in the mucosa. On the histology pictures, you can see a massive infiltration of immune cells, in particularly mononuclear immune cells in the mucosa and submucosa. In severe cases, we also have also the tunica muscularis is involved, and in these cases we have a transmural focal inflammation. Then to study the pathogenic mechanism and the involvement of immune cells, I performed flow cytometric analysis. Here you see the FACS results from the PAC IL-10 knockout mice, the interleukin 10 knockout mice controls, and the wild type controls. And you can see that the model has a pronounced increasement in the percentage of neutrophil granulocytes and also monocytes compared to wild types. On the other hand, there is a decreasement of the percentage of B cells, both of the PAC IL-10 knockout mice and the IL-10 knockout mice compared to wild type. Then I looked at the cytokines in the colon, and here you see 3 IBD relevant cytokines: TNF-alpha, interferon gamma, and IL-17. And you can see that these cytokines is significantly increased in the model during disease compared to the 2 control cohorts. These data, among other data, indicates that neutrophils activated macrophages as well as Th1 and Th17 cells could be drivers of inflammation in this model. Then I wanted to study the role of gut bacteria in development of disease of the model. As you all know, we have billions of bacteria in the gut, but something goes wrong in IBD patients. They have a loss of tolerance and an excessive immune response against the common cell bacteria. To study this in the model, I treated a cohort of mice with ampicillin, which is a broad-spectrum antibiotic. And here you see that treatment with ampicillin prevented weight loss in the model. The mice maintained the weight throughout the whole study. Over here you see the colonic weight length ratio, which is an indicator of inflammation in the gut. An inflamed colon gets thickened and shortened and therefore have a higher weight length ratio. You can see that ampicillin treatment significantly decreases the weight length ratio and therefore the inflammation in the gut. Last, I wanted to look at the predictive value of the model, and therefore I treated the model with known therapeutics against IBD. In this case, I treated the mice with anti-IL-12 23p4T, which is right now in clinical trial, phase 3, and it shows promising results. I had 2 setups, a prophylactic treatment, where I treated the mice from day 0 of the experiment, and an interventive treatment where I treated the mice from day 10 of the experiment. Here you see the weight length ratio and the histology score. And you can see that both prophylactic treatment and interventive treatment, the round spots, shows significantly decreases the disease progression in the model. So to sum up, the PAC IL-10 knockout model has a heterogeneous pathogenesis in which both the innate immune system and the adaptive immune system is involved. The model fulfills the 3-factor hypothesis, since the development of disease requires a dysregulated immune response, a decreased mucosal barrier, and the presence of bacteria in the gut lumen. The model develops clinical manifestation which mimics what we see in human IBD patients, and reference work shows predictive validity in the model. Also very important, this model is feasible since it produces an immediate reproducible response. So, all things considered, this model could indeed be a valuable in vivo model of preclinical studies of human IBD. Last, I want to thank my two supervisors, Thomas Lindebo-Holm from Novo Nordisk and Professor Axel Kornhoff from University of Copenhagen. I would also like to thank Peter Quist from Department of Histology, and of course my funding of the project, Novo Nordisk Life in Vivo Pharmacology Center, and I would like to thank you for listening. Thank you.